Okay, we're going to go ahead and get started. Good morning, everyone. Good morning and good afternoon to those joining us abroad. My name is Cassie Maximin from the IQ Secretariat. Thank you for joining us for today's webinar, Developing Predictive Models by Sharing Predictions. Today's webinar is presented by the In Silico ADME Working Group in coordination with the Translational and ADME Sciences Leadership Group. We are pleased to welcome you to this webinar sponsored by the IQ Consortium. The IQ Consortium was established in 2010 as a technically focused, not-for-profit organization comprised of pharmaceutical and biotechnology companies. Our vision is to advance innovative solutions to biomedical problems and enable pharmaceutical companies to bring quality medicines to patients. Our mission is to advance science and technology to augment the capability of member companies to bring transformational solutions that benefit patients, regulators, and the R&D community. Before we introduce our speakers, a couple of housekeeping items. All attendees will remain muted. For today's panel, the chat function has also been disabled. However, you may submit questions during and after the presentation using the Q&A panel found at the bottom of your screen along your toolbar by selecting the More tab. Questions will be addressed by our presenters at the end of the presentation, time permitting. Following this webinar, for inquiries regarding membership or this presentation, we welcome you to contact the IQ Secretariat. You may view a recording of this presentation and past webinars by visiting our IQ Consortium YouTube channel. Thank you. I will now hand it off to our moderator, Prashant Desai with Genentech, to introduce our speaker.
Thank you, Cassie. Welcome everyone for the webinar. And Raj and I, we are very happy to share the summary of our IQ project on student-teacher federated learning approach. And before you go into that, there is a disclosure for our respective employment with Genentech and Vertex. Next slide, please. So before we jump into today's main topic, we would like to give you a brief overview on the In Silico ADME Working Group within the IQ Consortium. The group was established back in 2015, so we've been working together for over the last 10 plus years. And over the years, we had participation from 21 different pharma companies, of which 11 of us have been actively engaged in research work. And over the course, we have published three papers to share our learning with the scientific community. And our main goal for the working group has been to learn and apply some of the best practices across the industry when it comes to use of in silico ADME model to advance our drug discovery portfolio, and also share this learning with the broader community to benefit the science as a whole. Going to the next slide, please. So over the course of this journey, here are the key milestones that our group was able to walk through. So starting from 2015, initially over the first year, we had exchange of understanding of our respective internal tools and what kind of challenges each of the companies engaged in this work were facing. And that really helped us shape up a white paper that provided overall understanding and learning of how in silico ADME models are used in industry, what are the current challenges, and what would the future for this field look like as the drug discovery advances. And that was published back in 2017 in JMedChem. And our next project was a research-based project where we systematically compared both in silico and in vitro methods to predict fraction unbound in incubation, specifically in hepatocyte and microsome. It's one of the important properties that is used in understanding how in vitro measurements of intrinsic clearance would translate to in vivo prediction or IVIVE, as many of you would know. And that work was published in Molecular Pharmaceutics back in 2019. And then we also had opportunity to actually host ACS Symposium in 2019 at the national meeting. And finally, we are today going to present the work on the student-teacher machine learning model approach for rat volume distribution. This work has just been published in JMedChem. So the paper would provide a lot more details than what we would cover in the seminar today. With that background, I will hand over to Raj, who will then walk through our learning and how the model was built, etc. Next slide.
Okay, great. Thank you, Prashant. So today we're going to be talking about how to build better predictive models. And I think it is commonly accepted that to build predictive models, you need the appropriate methods and you also need data. And in general, to get better predictive models, you need better data, which can mean more data, but also data of the right quality and with the right features. The sort of common thread, especially with more recent developments in AI, is that if you can get more and more data, then your predictive ability increases. Next slide, please. But the challenge is that data costs money, and some types of data costs more money. And as we probably know, some endpoints are very expensive to measure, things like volume of distribution, clearance, and so on. And essentially, if we could get more of these data sets, we could potentially build models that have a broader domain of applicability, but also with that extra data, the overall predictive accuracy would potentially be improved. By enabling this to happen, we potentially could get more generalizable models that would see a broader use of PK predictions earlier on in the design cycle. And so really the challenge is, how do we get access to more of these data sets? Next slide, please. So the problem statement that the in silico ADME group sort of decided to work on was stated here. How can multiple companies collaborate to build a predictive model for an endpoint that is trained on private internal data from all the companies without sharing any of that internal data or any of the internal methods? Next slide, please. And so this problem statement is essentially solved by the field called federated learning. Essentially, data and potentially methods are distributed amongst multiple participants, and some method is applied to combine, integrate, or aggregate the data and the methods across these participants without having to share anything explicitly. You click again. And a number of groups have been working on this. The Melody Consortium in Europe has been sort of working on this for a number of years and published their results a few years ago. More recently, Aferis has initiated a program on federated learning, primarily focusing on protein structure prediction. And LASA as well has been working on this problem for a few years. And later on in the slides, we will mention some comparisons between LASA, the work from LASA and our work here. You click again. But it's important to realize this is not a new problem. In fact, it goes back about 20 years. And I've highlighted a number of papers back from 2005, where a number of workers had been tackling the problem of trying to share data without revealing any sensitive information. And a variety of methods have been proposed. And over the years, this problem has been tackled more broadly in the general machine learning community. If you click again. And as a result, today's work is going to describe, or will be based on, a method called the student-teacher modeling approach. The more technical title for this approach is described or shown here in the screenshot from the paper that was published by Google about nearly 10 years back. Click again. And so this slide highlights the overall sort of conceptual workflow of what a student-teacher model workflow is. It starts off with a public data set. In this case, we use Campbell, where we simply take a selection of structures. In this case, it's about 130,000 structures. Those structures are then provided to participants. In our case, it was 11 participants, members of the working group, who then used their internal models for a particular endpoint to make predictions of those Campbell structures. And these lead, so each participant generates a set of predictions. These are then passed to the IQ organization, who anonymizes them and sends those anonymized predictions back to the working group. And using those predictions, we are able to build what we call a student model. Importantly, these predictions, anonymized predictions shown here, are also available to any participant within the working group to build their own models if they so wish. But the rest of this talk is going to focus on the student model that we developed as a group and that was included in the publication that Prashant mentioned. If you click again. So as you'll note here, the IQ organization plays a critical role in enabling this to happen. Essentially, it acts as an honest broker. During the planning of this project, we agreed upon pre-agreed upon a CSV format by using which predictions would be shared. And so each participant made predictions using their internal models and sent the resultant CSV in that format to the IQ person who agreed to play the role of the honest broker. So any identifiers that might reveal the company name or any other identifying information was made sure to be cleaned out and removed from those CSV files. And IQ then shared back the anonymized files to the participants. If any company had a question about the data, they were sent to the IQ person who proxied those questions to the relevant representative from that company. Click again. And so I sort of redid that previous workflow to show a little more detail of how the sort of data and predictions flowed through this that ultimately resulted in a student model. So again, as we start off with a 130,000 subset from Campbell that went through a cleaning and normalization process that was, again, agreed upon by the participants upfront. That was provided to IQ, who then essentially distributed that Campbell subset to the participants of this project. And importantly, the Campbell subset is simply a set of structures and their Campbell IDs and nothing else. The participants used their internal teacher models to make predictions on an endpoint that was pre-agreed upon by the group before this project started. And those predictions were sent back to the IQ representative. As I mentioned, the IQ representative made sure that any identifying information, such as company names and a file name, etc., were removed, and essentially provided back the Campbell structures, the Campbell IDs, and the predicted value. Essentially, we got 11 files or CSV files from all the participants, and these were provided back to the group. I'll go into a little more detail on model building, but we essentially aggregated the 11 predictions for each Campbell compound. That represents our internal training set, and that went into the model building process that resulted in a student model, which we then used to validate it using an external test set. Importantly, click again, please. Throughout this workflow, no proprietary chemical structures or proprietary assay data or any proprietary model was shared at any point. So none of the participants had access to any of the data of the modules from any of the other participants. Click again. So I'll hand over to Prashant to talk a little bit about the endpoint we chose to work with.
Yeah, so as Raj mentioned, we kind of discussed various options for which endpoint would be appropriate as a proof of concept for evaluating this approach within the consortium. And we settled on rat volume distribution for some of the reasons as I listed here. It's one of the low throughput resource intensive endpoint. And obviously that means that most companies will have a relatively smaller data set for such in vivo endpoint compared to in vitro ADME endpoints like microsome clearance. And then the other aspect is typically the way we measure volume distribution in PK studies are generally consistent across different pharma companies. So it will also reduce the concerns about data harmonization that are behind the model that each company has built. And this is the endpoint where we had maximum interest and participation from 11 different companies, and which kind of help us settle on this as the endpoint to go after. And finally, but last but not the least, it also supports the 3R principle. Having a predictive model for such endpoint should help reduce the use of animals during the drug discovery process. Next slide, please. And so this plot shows the initial input that we received from 11 different companies or teacher models. And by the way, we also wanted to ensure that any camel entries that were used as part of student training set, those compounds were excluded from the respective teacher models if they were part of it. And also the Lombardo test set that we use as a reference test set for evaluating the models, those compounds are also excluded to make sure that there is no contamination. Of course, there might be similar compound, but we did not go through the rigor of eliminating similar compound, but at least the exact match were eliminated. And what we noticed, what basically overall range of predicted volume distribution was pretty wide, ranging all the way from almost 0 to 1,500 liters per kg. We had, just because of how machine learning models work, we had a few predicted volume distribution which were negative, so we removed those predictions. Majority of molecules did receive predictions from all level participants, and so we had a decent size of 133,000 Campbell structures with 11 predictions for each of the structures. And we basically utilize a geometric mean of all the predicted values to arrive at the final value that will be used as a label for training the student model. Next slide, I'll hand over to Raj.
Yeah. So this slide sort of provides some diagnostics that highlight the fact that the external test set, in this case the Lombardo data set, is roughly similar to the training set, such that the validation experiments would be reasonable. In terms of the log p value, the test set, the Lombardo set, is shifted a little to lower values than the training set, but overall for all the other molecular descriptors, they sort of occupy more or less the same distribution. And you can see in this TSNE plot on the top left that the Lombardo data set, which are the blue points, are somewhat evenly distributed across the background of the training set. And so this gives us a degree of confidence in the validation results that we'll show a little later. Next slide, please. Before going into some of the results from the model itself, just a few points about the model. We employed a boosting algorithm using fingerprints and molecular descriptors implemented in RDKit. We didn't do any extensive hyperparameter optimization and went with the default settings. We did employ internal cross-validation, and that model is also available as part of the publication. So coming to some of the results of the model itself, this plot used an internal test set. So essentially a single random split of the 133,000 Campbell molecules into a training and test set. And the key point that we wanted to highlight here is that the student model shown, or the RMSE of the student model that's shown in red here, is lower than the RMSE from any of the individual teacher models. This is very encouraging because it does show that, first of all, by aggregating the predictions across multiple student models, multiple teacher models, the student model is able to show improved performance. This is also in line with previous work, especially from LASA, that has also shown similar behavior. The caveat here is obviously that this is one single test set and doesn't necessarily generalize, but it does serve to highlight the underlying methodology. Makes sense and works. You click again. Given that we have 11 predictions available to use, the final student model used all 11 predictions, but it was also interesting to ask the question of what would happen if we used, say, 10 predictions or 9 predictions, etc. And what this plot shows is the performance of the student model using different numbers of teachers. So and again, this is based on a single random test set. So this is more about exploring the methodology itself rather than making a claim about the prospective utility or prospective predictive ability of the model. But it is interesting to note that if you take a single teacher and treat that as the student model, the test set RMSE occupies a wide range, which again is not surprising because the individual participants had models of varying degrees of accuracy. The boxes correspond to the fact that if you take two teachers, there are two C11 possible pairs of teacher predictions that you can use to build a student model. And again, you see a reasonably wide spread of performance for all those models. But importantly, as you increase the number of teachers, there is essentially a monotonic increase in performance, that is lower test set RMSEs, as you include more and more teacher predictions. And the last point here corresponding to using all 11 predictions, and therefore there is a single model for that. And in general, some teacher models still do perform the student model, but the general trend is that the student model outperforms a few of the individual teacher models. Another question that did come up during the development of the model was that is a particular teacher model really driving the ability of or driving the performance of the student model? And to test that out, we essentially used a leave one out procedure where we first of all used all 11 predictions to build the student model, which is shown in this red bar here. And again, using a random test set, a single random test set. And then we rebuilt the student model, but leaving out one company's predictions. So the remaining bars correspond to student models built using 10 predictions, and each column corresponds to the company that was left out. And you can see here that there are some variations in the RMSE, but those are probably occurring at the second or third decimal place. Overall, our conclusion is that no single teacher model is driving the performance of the student model. If you go through again. So those are useful questions to ask of the underlying methodology, but the real question is, does it work in a prospective manner? And these two plots present those results, which report the performance on the external Lombardo test set, where we report the R squared of the test set predictions on the left-hand side, and the RMSE of those predictions. In both cases, the Lombardo dataset contains 329 compounds, and as Prashant previously mentioned, there is no overlap in terms of exact match between this external test set and the training set. The error bars are generated using a bootstrap method, and the take-home message from here is that the student model is not the best performing model. Neither is it the worst performing model, as you can see from the sort of position of the student model bars in these two plots. At the same time, if you look at the range of R squares and range of RMSEs, there isn't a very wide range. And in many cases, one could argue that, for example, an RMSE of maybe 0.52 is not all that different from an RMSE of, say, 0.49. And given the overlap of these bars, one could further argue that there isn't a statistically significant difference. So this was very encouraging that going through this process, we are able to identify or generate a model that does show improved performance compared to some individual teachers. Next slide, please. We also looked at the percentage within X-fold metric, which is a common way to use predictions for this particular endpoint. Again, this is the performance using the external Lombardo test set. So each teacher model was analyzed, categorized its predictions into either twofold, threefold, or within fivefold, and similarly for the student data set here. And again, you can see that the numbers aren't too different across all the individual teacher models as well as the student model. And in fact, within fivefold, the student model is actually one of the top three performing models. Next slide, please. Hand over to Prashant to go into some details about the compounds.
Yeah, so next we try to understand, at least based on the one test set, the Lombardo test set, what one would expect of the performance of the student model depending on the chemical space or type of compound properties, because that could help inform when would this model be more reliable versus not. Of course, one has to remember that this is based on the single external test set that we could access in public domain. In this case, when you looked at this in different ways, one thing was clear that for beyond rule of 5 compound, we saw a differentiation of performance versus compound within rule of 5, as you can see from the pie charts. above that compounds within rule of 5 generally had a better predictive performance for that set. And combine this with polarity, we use the TPSA, and using a cutoff of 140, we also noticed that for more polar compounds, in this case more than 140 TPSA, the performance was relatively poor compared to those with less polar. And in fact, when you combine the two properties, if you look at the 4 pie charts on the right-hand side, the top right corner seems to be the worst case scenario, where the TPSA of more than 140 for beyond rule of 5 compounds, only 39% of the compounds were predicted within 3-4 by the student model. Whereas the other bookend is on the bottom left corner, where compounds within rule of 5 and having a TPSA less than 140, 70% of those were predicted within 3-4. Next slide, please. Also looking at this from the acid-base point of view, we had somewhat decent distribution of base and neutral, relatively smaller number of acids and deuterion. Of course, not surprising. These are basically drug-like molecules from public domain, so we would expect that. But comparing the model performance across these different protonation, what we noticed was acids seem to be the most difficult compound to predict by the student model. This likely has to do with the representation in the training set of the teacher model, but we did not necessarily went back and confirm that, but that's just our hypothesis. And then deuterion somehow, although the n is small, deuterion did show the lowest average prediction error, followed by the bases and the neutral compounds. Next slide, please. Raj will take over.
So at this point, I think what we've been able to show is that the method works. It does lead to a model that is useful. It does perform better than at least some of the individual teacher models, both in terms of internal validation, but also in terms of prospective utility. The next question that we asked was, can this be more automated? So as I mentioned previously, this depends on having an honest broker, in which case, in this case, it was the IQ Secretariat. That was sort of a natural role for the IQ Secretariat to play because obviously the in silico working group is part of IQ. But it turns out that once the participants had agreed on the format of the teacher predictions, it is feasible to replace the role of IQ as a broker with software and essentially potentially run this autonomously. Now, this still does mean that the software needs to be run on a server owned by some party, so there's still a need for a neutral trusted organization to operate that software. But given that, it does reduce the reliance on having an actual person be responsible for sort of playing the role of the honest broker. Next slide, please. Okay, so what are the next steps from here? So one of the things that I think really caught our eye, at least when we were planning this project, is that based on the description in the original Google paper, the workflow should be relatively straightforward. And it actually did turn out to be that this workflow for federated learning is very simple in terms of logistics. It is low cost and it is model agnostic. And the fact that we are describing RAT VDSS here, or models for RAT VDSS here, doesn't preclude using this workflow for any other endpoint. Really, the bulk of the effort here was to reach a consensus within the in silico working group to decide what is the endpoint that we want to measure, and that would be useful to test this idea. But once that was decided, it was a little more discussion to discuss how predictions should be shared and some discussion about how the model should be built. And beyond that, as well as the role of IQ here, there is no need for any other infrastructure investment or development to implement this workflow. We've shown that we've established a strong baseline for a machine learning model for log VDSS. But as I mentioned, we haven't done hyperparameter optimization. We explored a little bit of the descriptor space, but there are a lot of opportunities for further optimization to achieve something that's more state of the art. And the paper that has been published includes the aggregated Campbell data set for the community to explore. The code and the data is also available in a public GitHub repository. Good. Next slide, please. So I think there was one slide we skipped. If we could go back one more. Yes. The concerns and considerations. So one of the major topics that we did discuss and spend a lot of time discussing was the potential for information leakage. And we have in the paper, we've spent a decent amount of effort explaining potential sort of attack vectors by which a bad actor might be able to extract information. And we feel that this approach is relatively safe. If you are interested in more sort of mathematical foundations, the Google paper actually provides mathematical proofs of the security of this method. But in practical terms, there are still some considerations that it's useful to be aware of. So in terms of the data, the teacher models are obviously built internally by different companies. They use different data sets, potentially using different assay protocols. As Prashant mentioned up front, the choice of Vdss in this case was sort of driven by the fact that the results obtained for the different protocols don't vary significantly, and thus we sort of avoided the need to explicitly harmonize data for this case. That may not be the case for other endpoints where assay protocols might lead to substantially different readouts that will require explicit harmonization or may not be able to be used together. The other aspect that is going to be familiar for people used to developing machine learning models is that the teacher predictions include noise, and that is going to be incorporated into the student model as well. And as I mentioned previously, some endpoints may require alignment and harmonization efforts across the participants. As I started out, security is obviously a concern. Can information about the teacher models in the training sets be inferred from the student model? And as we discussed in the paper, we feel that it is extremely unlikely that any information about the training sets from any given company, their models, can be inferred from the student models. So we feel that this is a very secure approach to federated learning. Finally, I opened this talk sort of suggesting that one reason to do this is to get more data to increase the applicability domain of the model and therefore make it more generalizable. Unfortunately, given the constraints that we're operating under, in that nobody obviously wants to share their training sets, it's unclear if this method actually leads to an expanded applicability domain for the student model. Keep in mind that the student model is essentially trained on the Campbell dataset, and so the applicable domain of the model is essentially that of the Campbell dataset. Now, expanding or comparing that training set to the training sets for the individual companies is something that we couldn't do. And so this question, I would say, remains a little open. And I think any individual company is able to take the training set and do their comparison internally and reach a conclusion. But this is not something that we as a group would be able to conclude. So if you go to the next slide, and pick one more. And so in closing, it turns out that the actual model development was relatively straightforward. We used off-the-shelf methods for the model, off-the-shelf methods for the descriptors, and a pretty standard workflow. I think the real challenge is determining on what information would be included in the predictions, how predictions would be made, whether we should include, say, confidence values of the predictions, as well as addressing concerns around data leakage. Ultimately, we all benefited from the IQ legal framework within which all the participants operated. That made it a lot easier for individual companies to agree to share predictions on the Campbell dataset, as well as, of course, IQ playing the role of the honest broker in this workflow. Next slide, please. So I'd like to acknowledge all the participants who engaged in discussion and planning of this project, provided the predictions, and contributed to the publication. And thank you for your time. Happy to take questions.
Yeah, I think there are questions, a couple of questions are coming in. Let's start with something that you already touched upon, Raj. So this question is about different types of data. In this case, we used rat volume distribution where we had general consensus that it should be um, more or less similar across different companies. But the question is about what about parameters where they are more assay dependent, something like solubility or Keiko2 permeability, etc. How do you approach data harmonization? So what are the thoughts on handling those kind of endpoints for student-teacher model approach?
Right. So I think definitely for some endpoints, it will not be as straightforward to just sort of merge predictions from data sets from models built on data sets from very different assay protocols. I think in that case, there would have to be an initial step before sort of starting off such a project where participants would need to at least disclose the nature of their assays and be able to then decide whether some participants who are using very different assay protocols simply would not be able to contribute predictions. The other aspect is also to note is that there is literature in the field of federated learning in the life sciences where people have tackled this question. There seems to be some consensus that for a variety of federated learning approaches, it doesn't matter so much. I'm personally not entirely convinced about that. I mean, it's easy to treat these predictions as numbers and then merge them and sort of ignore the underlying biology or chemistry that is sort of generating the data. So I do think that before embarking on say another project like this with a different endpoint, a discussion of the underlying assay technologies is going to be necessary, and that will drive which internal models can actually be used for this workflow.
Yeah, and just to add to that, I think as Raj mentioned, if there is enough interest to still try to tackle that problem, one surrogate approach could be if most companies typically would use known drug molecules or reference compounds in the respective assays. So one could compare the outcome for those compounds which are tested across these different protocols. And while we may not expect exact one-to-one match, if we actually observe a relative rank order or even categorical separation that is reasonable across these different companies or different assays, in theory, a group could agree on using more of a categorical approach than a numerical approach and still kind of harmonize the data. But it will depend on what level of agreement or alignment you see even when you look at categorical separation. Because in the end, especially for in vitro endpoints, often when you apply in drug discovery programs, even a categorical model might still be helpful for the drug discovery teams when they are looking at large virtual screening and filtering molecules based on desirable property space. So one could try to approach it, but as Ras mentioned, it will get a little bit complicated depending on how different the assay protocols are. There is another question. This is about continuing this kind of work. So the question is there any interest from companies to continue this work, sharing data and building models for other agreed upon endpoints? I can speak to that. Actually, when we started this, that was our intent, that we'll start with one endpoint, and based on what the outcome is, if companies are really excited to continue this work, we could continue this project with additional endpoints. Unfortunately, given the nature of the IQ and the voluntary participation for all the companies, this project itself actually took some time to get things rolling, given that we are trying to work across 11 different companies, different time zones, different level of commitments for day-to-day job. So when we finish the work, we have kind of decided basically to sunset this IQ working group. So this is basically the last project that we tackled together. And so we won't be able to continue this through the IQ working group. But as Raj mentioned, I think this opens the door for others to come together and come up with similar agreement, given quote unquote how simple it is to, at least from technology point of view, it could be handled with a very simple technology to share data and build models of this kind. Any other thought, Raj, on that?
No, that's a great summary. Like as Jean said, it certainly it took effort to pull this together. And as I mentioned, a lot of that effort was in reaching consensus on how the thing would operate. But since this working group is sunsetting, I think there's still opportunities within IQ. Other groups could certainly take this workflow up, and I'd certainly be happy to help out and advise. But the in silico ADME working group will not be doing any more on this.
Yeah. Another question here. Will these ADME models are agnostic to therapeutic modality? For example, small molecule versus peptides versus antibodies or proteins? Or if not, are there plans to consider these modalities in future? So I think this has to do with what was under the hood for respective teacher models, because in the end, the Campbell predictions, the reliability of the Campbell prediction will obviously depend on what kind of training set coverage was provided by the individual teacher models. I think in this case, we did not obviously share those level of details about, as Raj said, that we needed minimum information to be shared about the respective training set. So we do not have a clear information about whether any given company's training set included just small molecule versus all kind of modalities were part of the training set. But if that was possible, I think one could get some qualitative estimation, some relative applicability domain, depending on the application, chemical space of the teacher training set, if the parties involved are willing to share such details. Next question for you, Raj. Should be straightforward. Does this model consider quantum mechanical descriptors?
No, it just used standard fingerprints and physchem molecular descriptors.
This one, let's see if we can figure out what it. This one says, are there opportunities to make this optimization process open source after blinding identifying information? I think if it is about the student data, that is obviously already shared. So the training set for the student model has been shared, and include even the frontline model that Raj described in detail is also available directly to use for those who want to just use that model. But I think there's a lot of opportunity to improve the model itself by just starting with the student training set and apply different modern machine learning approaches to improve the model itself. Any other thought, Raj, on that?
No, that's pretty much it. The data is available, and the model, and as well as the code to generate the model from the data in the GitHub repository is also available. So given that, anybody should be able to play around with it and build better models.
Yeah. The next one is interesting. Are you actively implementing this in your respective companies? So I think this is obviously going to be organization dependent. Obviously, as Raj said, each of the participant companies have now access to student models. So it is imperative that they could conduct more exhaustive evaluation of the student model versus their own teacher model against different test set, including their own internal programs to evaluate that. But as of now, we haven't heard or shared specifically any comparison of the student and teacher model within individual organization yet. But in theory, yes, it's obviously doable. I think, Raj, this is somewhat a philosophical question, but I'm sure you have some thought. What do you think is the biggest barrier to more widespread adoption or buy-in from companies for such federated learning approach?
So, I mean, I have a few thoughts about this. I mean, it's interesting that in the last maybe 2 or 3 years, federated learning has picked up. The Melody Consortium that I mentioned previously, I think sunsetted maybe a year or two ago. That was both an industry academia collaboration. But more recently, LASA, for example, runs a consortium for interested parties that employs a federated learning approach. And more recently, Aferis is a commercial organization that has rapidly grown in the number of participants who are sharing or using the Aferis approach to federated learning for protein structure prediction. And I guess another example recently is the Lilly Tune Labs effort. So I think there is an increasing recognition that there are benefits from federated learning. And I think also the sort of barriers are coming down by way of organizations like FRS or Lilly Tune Labs that are implementing infrastructure that provide both legal guarantees as well as security guarantees that proprietary data is not going to be shared or leaked and stuff like that. I think the barrier ultimately is going to be sort of around the risk of IP leakage. And I think as we move forward, the machine learning community is obviously coming up with methods and sort of more formal, sort of computer science driven or mathematically based guarantees of security that obviously doesn't map all the time to actual real world implications. And I think examples like Lilly Tune Labs and Aferis are showing that if the infrastructure is properly planned out, it is possible to do these and address the risks that parties might have. So I think the biggest barrier is risk of IP leakage, but at the same time, it does appear that that is being addressed successfully.
Yeah, I think another question that came up, and this is something we also discuss as a working group, is what can we tell more or do more about the applicability domain of these models? Of course, as part of this research work, we use the Lombardo test set, but that was the main reason was it was publicly available test set, so we could freely exchange it and work on it together. But that doesn't necessarily represent all the type of chemical space that an ongoing drug discovery program may be working on. So what are the ways that if somebody wants to use a student model on their actual drug discovery program, what would they do to kind of understand the applicability of these models? So I think in that case, obviously, like as is done typically for any machine learning model, when it's used in real time, the best way to understand its applicability domain is to learn and test. So one will have to conduct some measurements for the chemical space that they are interested in, and that would be the best way to understand whether the given model, in this case could be the student model, has a signal that they could utilize to understand the applicability for the respective chemical space that they are trying to evaluate. And in theory, one could actually enrich the student model further with their respective internal training set. So while the training set may be small, now is enhanced with the existing student model, and that could be another way to kind of to see if using them together would further improve the applicability domain for the chemical space that one is interested in. Okay. I think we have addressed all the questions that came up in the Q&A session. We have a few minutes, Raj, if you have any other thoughts that we would like to share with the community before we close.
I mean, at this point, obviously, I want to acknowledge the efforts from all the members of the In Silico ADME working group. This project did run for, I think, 4 years. And as Prashant noted, this is sort of extra work on top of everybody's day job. So I'm really grateful that everybody agreed to participate on this, as well as take the time out to sort of modify their internal models to exclude compounds from the training sets, et cetera, as Prashant mentioned, but also take the time to do their internal evaluations, contribute to the paper, and ultimately get us to where we are. And I want to reiterate as well that again, the machine learning field is coming up with many ways to do federated learning, and within the sort of pharma industry and broadly life sciences domain, we are seeing more and more examples of federated learning. Obviously, I'm biased, but I do think that the student-teacher model lends itself to very easy implementation on the technology side of things. And sort of that alleviates the sort of challenge of implementing something like this and allows participants just to focus on the data they want to model and how they should share that data between each other. And so overall, I feel in a way the ROI on this approach to student-teacher or federated learning generally is quite high.
Yep. And I would simply add that not only for this specific project, but the overall journey of the IQ working group, this in silico iron working group has been very fulfilling for all of us. It gave us a really good platform to exchange our scientific curiosity and also learn from each other's experiences on how things are working, not just on technical side, but also in terms of changing the culture, the mindset of how models are used across different pharma companies. And I think we have come a long way in the last 10 plus years that we have worked together. So I would like to take opportunity and use this platform to really thank all the members of the IQ working group. And I hope some of them are actually dialed in and here to celebrate this last successful project and basically hopefully the community will continue this kind of exchange and collaborations, and we would love to see more and more of such work being shared broadly for everybody to learn from. I think with this we will conclude the seminar. Thank you everybody for joining. And feel free to reach out to us offline if you would like to learn more about this or have more ideas and exchanging. We are open to communication. You can reach both of us on LinkedIn. So hope to hear more about this and see more of this work come out in literature.
Thank you.
Thank you.